Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 17 from our Campbell's 12th edition biology textbook. This chapter covers gene expression from gene to protein. Quite a fun chapter, so let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, this chapter is so interesting. It's all about genes and how genes code for proteins. Do you remember that? I had mentioned several times before that the purpose for genes is that usually genes code for proteins. So the insulin gene, for instance, would be the information that your pancreas would need in order to build the insulin protein. Does that make sense? Genes code for proteins, and the process by which your cells make proteins from that gene information is called gene expression. Does that make sense? So we're going to learn about gene expression. Now, before we delve into exactly how gene expression works, I just want to share with you why it's important to learn about gene expression. Everyone knows that genes give you traits, right? So if I asked you, what do you need genes for? You would say traits. But there's a whole bunch of missing components there, right? So how do you go from a gene to a trait? That's what this chapter is going to clear up for you. So just to give you an idea, a gene is a segment of DNA that codes for a particular protein. That segment of DNA is first copied into complementary mRNA or messenger RNA by a process known as transcription. And then the ribosomes will read that mRNA message through a process known as translation in order to produce a protein. That protein functions to give you your traits. So for instance, this is some kind of gene for a protein that makes a pigment. This pigment gives the brown phenotype for this animal. Now conversely, there might be an albino animal. How would the gene cause albinism? Well, look, that same gene, if mutated, right? Let's say this animal has the mutated version of this pigment gene. Well, then that gene would be copied again by transcription to form a messenger RNA. That messenger RNA would be translated into a non-functional protein. This protein may have folded incorrectly because there was a mutation on the gene somewhere. Well, anyway, if it's a misfolded protein, it's not going to function correctly. It's not going to produce the pigment. And if you don't produce the pigment, you're not going to have pigment in this animal. And this animal is going to lack pigmentation. Does that make sense? So yes, genes can lead to traits such as albinism. But now you know why. It's because during gene expression, proteins are made, and those proteins function or don't in order to give you your traits. Again, I know this is a little confusing because we haven't gone into what does transcription mean, what is mRNA, what does translation mean. We haven't gone into that, but we will. This is just kind of a preview, an overview, foreshadowing of what we're going to learn about. But it's very important to pay attention because without understanding gene expression, without understanding transcription and translation, you don't understand why genes give you your traits. And that's a very important thing to understand. So let's pay special attention and learn all the concepts so that you can understand why genes lead to traits. Okay, again, the DNA inherited by an organism leads to the traits by dictating the synthesis of proteins. The genes lead to synthesis of various proteins. Those proteins function to give you your traits. So because of that, proteins are the links between genotype and phenotype. And remember what this process is called? It's called gene expression. This is the process by which proteins are made from the gene information. So the process by which DNA directs protein synthesis. And remember from the previous slide, this includes 2 stages that we need to learn about. The first stage is called transcription. This is where the gene is copied into complementary RNA called mRNA. And then the second step is called translation. This is the step where the ribosomes read that information on the mRNA and use that information from the mRNA to produce the proteins. Remember we've talked about before how ribosomes synthesize proteins? Well, you're going to learn exactly why in this chapter. Now, the way the information flows during gene expression has a term associated with it that you need to know. It's called central dogma, the central dogma of molecular biology. And this central dogma states that information, heritable information, travels from DNA, your gene, to RNA, remember mRNA or messenger RNA, during transcription. You could call this first step transcription, in which DNA information is copied into RNA information. And then how does the information flow? From there, there's RNA is translated to protein, right? So the second step here, the second arrow would represent translation. The first arrow represents transcription, and the flow of genetic information from DNA gene, to messenger RNA, to protein is known as the central dogma of molecular biology. And this is how information flows. What you should realize, though, is this process of gene expression occurs differently, slightly differently, in prokaryotes. As you can see on the left, this image represents what's going on with gene expression in a prokaryote, and eukaryotes on the right. Okay, so let me just break this down for you because we're going to go into all kinds of detail, but I'm just showing you the overview of what we're learning here. In a prokaryote, remember, there is no nucleus, so the DNA, remember the single circularized chromosome? That DNA is floating around in the cytoplasm. So what you should realize is pretty much everything's just happening in the cytoplasm, right? Because the DNA is in the cytoplasm, the ribosomes are in the cytoplasm. So what you should realize is the entire process of gene expression occurs just in the cytoplasm because there's no nucleus, right? So what do? We have hundreds or thousands of genes on that circular chromosome, but we just want to copy the information for one gene, right? We want to express one gene. So all we do is we copy the information for that one gene into mRNA. What was that process called? Can you beat Wicket? Of course, you're right. Transcription, right? So we're going to copy a gene, and we're going to copy that gene to make complementary RNA called mRNA, which is short for messenger RNA. And then what's the next step? Have we made a protein yet? Can you beat Wicket? What's the next step? How do we make a protein from the message on the mRNA? Right, as always, translation. So ribosomes come along. You know what the ribosomes do? They actually chomp down on this mRNA and they read the message on the mRNA, and along the way they string together the correct amino acids to make that polypeptide, that chain that will lead to a protein. Remember what proteins are? Let's take a step back. Proteins, remember, are just a long chain of amino acids. Proteins are like a pearl necklace of amino acids. So that's what we're doing. The ribosome reads the mRNA message, which is simply a copy of the DNA, a complementary copy of the gene itself, and the ribosome just reads the information on the mRNA, and it knows, okay, I should put this amino acid first, and then this amino acid next to it, then this amino acid. It's simply reading through the mRNA to string together the proper polypeptide chain, the proper primary structure of the protein, right? And again, this is known as translation. So if I asked you, where does transcription and translation occur in a prokaryotic cell, what would you say? And can you beat Wicket? Right. as always. I hope you beat him. The transcription and translation occur in the cytoplasm in prokaryotic cells, right? And that makes sense. The DNA is in the cytoplasm, the ribosomes are in the cytoplasm, everything's in the cytoplasm. So transcription and translation, the entire process of gene expression occurs in the cytoplasm, right? And by the way, let me tell you something. This is a little bit jumping ahead, but I think you can follow along. Did you know that in a prokaryote, like a bacterial cell, translation, which is the second step, right? Translation can actually begin before transcription is even finished. Isn't that interesting? So look at this. If the DNA is being copied to mRNA, before you're even finished, okay, before you're finished making the entire mRNA, the ribosomes are so eager to chomp down and start reading the information to make a protein. So isn't that interesting? Before the mRNA is even complete, the translation is happening. Isn't that interesting? So because of that, you know what we say? That not only do transcription and translation occur at the same place in prokaryotes, but they happen roughly at the same time. They happen at the same time, same place. And why do we say same time? Well, because you do not need to wait for transcription to complete before beginning translation. Isn't that interesting? So we should remember that for the exam. Now, sticking with our overview, let's look at what happens in a eukaryotic cell on the right. Notice that the eukaryotic cell is much larger. It has what? Remember, eukaryotes have a nucleus. This purple structure is the nucleus. And do you guys remember that the DNA is found inside of the nucleus, right? So our chromosomes, let's say if this was a human cell, we'd have 46 chromosomes inside of the nucleus. And remember, each chromosome houses hundreds or thousands of genes. We only have 46 chromosomes, right? But we have like between 23 and 25,000 genes. So each chromosome houses many, many genes. But remember, during gene expression, we're not copying all the DNA into mRNA. We're just copying the gene we want. If we want to express the insulin gene, then we just copy the insulin gene. That's it. No other genes, no other DNA. Just copy the insulin gene to mRNA by a process known as transcription. And then, later on, that mRNA will be translated by the ribosomes into the insulin protein. Does that make sense? So during gene expression, we are only copying the gene we want, only the little piece of DNA that we need, right? So remember, our genes are inside of the nucleus. So where do you think transcription needs to occur in a eukaryotic cell? Can you beat Wicket? Yeah, obviously it makes sense, right? Good job, Wicket. So the transcription has to occur in the nucleus, doesn't it? Think about that. If the DNA is in the nucleus, does the DNA ever leave the nucleus? No, right? So the DNA is in the nucleus. So transcription must occur in the nucleus. That makes sense, right? That means that translation has to occur outside the nucleus, right, where the ribosomes are. Because remember, ribosomes, they don't live in the nucleus. So right here, you can already tell that transcription has to happen in the nucleus. Translation has to happen outside of the nucleus where the ribosomes live. Does that make sense? So that's in the cytoplasm. But do you notice there's a whole nother little step here? Let's focus on this real quick. Focus on this with me. It looks like the gene is copied during transcription. That makes sense, to make mRNA. But look what it's labeled here. I want you to focus on this. This is strange, right? Look at this. It's not just mRNA, is it? It's called pre-mRNA. What in the world does that mean? That means that the mRNA is not ready for translation. So we've copied the gene, but that copy, that mRNA needs processing, okay? And so we're going to learn this in a subsequent slide. We're going to learn that this mRNA needs to undergo a process called RNA processing. This RNA processing modifies the pre-mRNA to mature mRNA. Does that make sense? And look at this on the left real quick. Look at the bacterial cell. Was there any RNA processing that needed to occur? No. So what you should understand is that in bacteria and other prokaryotes, there is no RNA processing necessary. In fact, remember, translation can begin before transcription even finished, okay? But look at this on the right. In a eukaryote, we're more complicated. Okay, we need to do transcription. We need to make a copy of the gene as mRNA, but then we need to process that RNA into mature mRNA. And by the way, there are 3 modifications, and we're going to learn all this stuff soon, but there are 3 modifications we need to do to the pre-mRNA to make it mature RNA. One occurs at this end called the 5' end. One modification occurs at the other end called the 3' end. And then another modification occurs in the middle. Okay, so there are 3 big modifications that occur during RNA processing to make mature mRNA. Then what? Do you think at this point we're ready for translation, right? So guess what? Again, I told you the DNA doesn't leave the nucleus, so the mRNA will leave the nucleus and meet up with who? Meets up with the ribosomes. Remember, the ribosomes chomp down on the mRNA at one end. And by the way, I'll just share this with you. The ribosomes attach at the 5' end on the mRNA, somewhere close to it. And then they read till they get closer to the 3' end. Does that make sense? And what are they reading? They're reading that genetic code on the RNA. They're reading that genetic code in order to do what? In order to string the correct amino acids together to make the polypeptide chain, to make that protein for you. Isn't that interesting? So again, I just want to share the takeaways of this slide. This is an important overview slide. This is what I need you to know. Are you ready? I'll give you a chance to bring out your notes. Okay, so in a prokaryotic cell, like a bacteria, transcription and translation occur at the same time, same place. That place is the cytoplasm, and they occur at the same time. You know why it occurs at the same time? Because no RNA processing is necessary, and everything's happening in the cytoplasm, so why not, right? Now, juxtapose that with the eukaryotic cell contender on the right. Okay, you've got your genes, you got your DNA. Now remember, the DNA is inside of the nucleus. Therefore, I need you to know that transcription occurs in the nucleus. And then, are you ready for translation right away? No, remember, in eukaryotes, after transcription, we have a copy of the gene, an RNA copy of the gene, but it's called pre-mRNA because it needs those 3 modifications. One modification at the 5' end, one modification at the 3' end, and some modifications in the middle. And that process is known as RNA processing. And so that mRNA then leaves the nucleus, meets up with the ribosomes for translation. So what I need you to know about the eukaryotic cell is that transcription and translation neither occur in the same place, nor do they occur at the same time. Does that make sense? Transcription occurs in the nucleus first, and it needs to be completed, and then RNA processing. Then translation can occur in the cytoplasm later on, well later, right? So does that make sense? So hopefully you see the big picture overview. You know how I like to show you guys big picture overviews before we go into great detail? This is your big picture overview slide. So we can keep referring back to this slide to see, where we're at in the details before we delve into those finer details. Does that make sense? But I hope this made sense, because now we're going to go into more detail. Now let's go into a little more detail about transcription, looking at how the DNA is copied into mRNA. Remember, we're not copying all the DNA, we're just copying one gene. So, this right here, let's look at what we're analyzing here. This is a gene. right here you have one gene, and by the way, the DNA would extend for miles and miles this way to the left and miles this way to the right, because remember, each chromosome contains hundreds or thousands of genes, right? But we're just looking at one gene, one piece of, one segment of the chromosome. Now, don't we need to copy this gene? We need to copy the information. And so to do that, this enzyme needs to sit down on the DNA. This enzyme that looks like a guitar pick right here, okay? This enzyme is called RNA Polymerase, not the one that we talked about in the previous chapter. We're not talking about primase. We're talking about its cousin, RNA Polymerase. RNA Polymerase attaches to the DNA in front of the gene. You see this here in dark blue? This is known as the Transcription Unit. This is what the RNA polymerase is going to copy. Does that make sense? But in order to copy the transcription unit, first RNA polymerase attaches to the DNA in front of the gene, upstream. It's called upstream of the gene. And it attaches to this green segment of DNA. This green segment of DNA has special sequences on it known as the promoter, okay? So we should know that the promoter is simply a stretch of DNA with a special sequence on it that attracts the RNA polymerase. And this RNA polymerase attaches to the promoter sequence, which is in front of the transcription unit, which is the part of the gene that needs to be copied. Does that make sense? So again, the RNA polymerase will attach to the promoter. Then the RNA polymerase will separate the two strands of DNA. Look at this. So RNA polymerase knows how to separate the two strands of DNA. It kind of like a helicase. Remember helicase? Helicase can unwind or unzip the DNA. Well, this RNA polymerase can unwind and unzip the DNA. And do you remember that RNA polymerase does not need to, it can start a new strand of RNA. It doesn't need anything to build off of like DNA polymerase 3 does. So you see what it did? Look at this. Look at this closely with me. Okay, look. RNA polymerase attached to the promoter sequence in front of the gene. RNA polymerase will then separate these two strands of DNA, you see? And then it will move to the right. But look at this. RNA polymerase will not start copying the DNA until it reaches this special spot called the start point. Okay, this is the start point. This is a point, a special location in front of the promoter. Okay, so only after the start point does transcription begin. The RNA polymerase will start copying the DNA to complementary RNA at the start point. And notice, I want you to notice something, and I want to see if you can beat Wicket based on what you see here. Does it look like the RNA polymerase is copying both strands of DNA, or does it look like it's only copying one strand of DNA? Can you beat Wicket? Right, as always, Wicket. It looks like we're only copying the bottom strand. And so the strand that you're copying is known as the template strand. Check this out. Zoom out for a second. We're only copying the template strand of DNA. Okay, and we're copy. And by the way, in case you're wondering, yes, we are building the RNA 5' to 3', because remember, both DNA and RNA are synthesized in a 5' to 3' direction. So we are building the RNA 5' to 3', and we are only copying the template strand. We leave the other non-template strand alone. Does that make sense? And do you know what we do now? We just continue on. This is called elongation. The RNA polymerase will keep going. Okay, let's see what's going on. As the RNA polymerase, let's see here, as the RNA polymerase proceeds, the DNA rewinds, right? Rewound DNA. And this right here is known as the transcription bubble. And notice how the transcription bubble will proceed to the right. We just carry on, right? Just carry on, unzipping, 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 and this little transcription bubble will carry on and on and on. And the more you copy, the longer the mRNA transcript becomes. And notice how you're building the mRNA transcript 5' to 3'. Isn't that neat? So we copy, copy, copy. Okay, and again, notice how we are only copying the template strand of DNA. We are not copying the non-template strand of DNA at all. Okay, and again, we also say that the RNA polymerase is traveling downstream. The promoter in green was upstream. That's where RNA polymerase attached. Then RNA polymerase began transcription, copying the DNA to mRNA on the template strand at the start point. And then it just carries on and on. That's called elongation. And the more the little RNA polymerase travels, the longer the RNA transcript becomes, because we're copying the DNA to mRNA. Does that make sense? So we copy, copy, copy. And then at some point, look at this. We have copied to the end of the gene. Again, we're not trying to copy everything. We're just trying to copy the gene, the transcript unit, right? And that is this dark blue region. Once you have copied this dark blue region, then you have completed your RNA transcript. And the RNA polymerase lets go, and the mRNA is complete. And notice this is the 5' end of the mRNA, messenger RNA, and the 3' end of the messenger RNA is on the other end. And look what this is. Again, this would be called mRNA. And this mRNA is simply a complementary copy of the template strand of the gene. Okay, so hopefully that made sense. This is the process of gene transcription. Here you can see the portion of it called elongation. Remember, the promoter was where the RNA polymerase attached. That was upstream of the transcription unit of what you want to copy. Then the RNA polymerase was able to unzip or unwind the DNA, move to the start site, and then start copying the DNA to mRNA. And we're only copying the template strand, synthesizing a new RNA transcript 5' to 3' until we reach the termination site somewhere around here. And then RNA polymerase lets go, revealing the newly synthesized mRNA transcript. Isn't that neat? So this is essentially what you need to know about transcription. This is how transcription works. We need to read the DNA to copy it into mRNA. And the mRNA is just a copy of the DNA, okay? But of course it's RNA, right? So if you see an A, you're putting down a U, right? If you see adenine, you're putting down a uracil, right? Instead of a thymine, because you're copying the DNA to mRNA, right? And again, this is known as transcription. Here's a closer look. If this is your guitar pick friend, RNA polymerase, this must be the transcription bubble where the DNA has been unwound. And here's your mRNA being synthesized. Notice this, you're building mRNA. So if you see a T, you put down a complementary A. But if you read an A on the template strand of DNA, you put down a U. If you see a G, you put down a C. G, you put down a C. T, you put down an A. T, you put down an A. But remember, for complementing A's, you have to put down a U. And notice again how we're building the newly synthesized mRNA is being constructed from a 5' to a 3' direction. Isn't that neat? Here's a closer look at the promoter. I just want to show you how the promoter works in a eukaryotic cell. Remember, this would be at the very beginning of what we just explained here. This would be during initiation of transcription. Remember I said at the very beginning, your guitar pick looking friend here, the RNA polymerase attaches to this green area called the promoter, which I said contains a special sequence. So let's take a close look at this right here. I just want to show you a little bit more detail here and see what we can learn. So take a look. This is a close look at that green area, the promoter. Again, this is looking at a eukaryotic promoter. Eukaryotes have a special sequence in that green area, in that promoter, called the TATA Box. And the reason it's called the TATA Box is because the sequence is TATA, TATA. And this is the sequence that's recognized by the RNA polymerase. Okay? Now, what's interesting is that in eukaryotes, other proteins must attach to the promoter first. These proteins are called transcription factors, and only then is RNA polymerase 2, it's called RNA polymerase 2 in eukaryotes. That's when RNA polymerase 2 is recruited to the promoter. And do you remember what RNA polymerase 2 does? RNA polymerase 2 attaches to the promoter, and then it separates the two strands of DNA. See this little bubble forming? That's the transcription bubble forming. The DNA is separated, and at the start point, that is where transcription starts, right? So if I asked on the exam, let me ask you this, can you beat Wicket? Is the promoter itself transcribed into mRNA? No, Wicket, that's great, good job. We're not trying to copy the promoter. We're not trying to transcribe the promoter. The promoter is this green area where the RNA polymerase attaches upstream of the transcriptional unit, upstream of the gene. The promoter is for binding of the RNA polymerase. Where do we start copying the DNA? We start copying the DNA at the start point. There's a special sequence here, the start point, where the DNA is copied into RNA. The DNA is copied into messenger RNA. Isn't that neat? And again, as I mentioned earlier, please realize that in prokaryotes, these little proteins don't need to attach first before RNA polymerase can attach. The RNA polymerase attaches directly to the promoter in prokaryotes, but in eukaryotes, other proteins need to attach to the promoter first. These are known as transcription factors, and then RNA polymerase can attach. But that's the minor difference that you need to know about prokaryotes and eukaryotes. Students often like to know too, they're very curious about how fast transcription happens. And it says right here from your textbook that transcription progresses at a rate of 40 nucleotides per second in eukaryotes. So you're copying 40 A's, G's, C's, and T's a second in the transcription process. That's how fast transcription is occurring. And how does termination work, right? We talked about initiation. This is where the RNA polymerase attaches to the promoter and separates the two strands of DNA and starts transcribing the DNA into mRNA at the start point. We talked a little bit about elongation, you know, how we're going at 40 nucleotides per second and we're copying that DNA in a 5' to 3' fashion. But how does termination work? How does transcription terminate? Well, as always, the mechanisms of termination are different in bacteria, prokaryotes, versus eukaryotes. In bacteria, The polymerase stops transcription at the end of the terminator. The terminator is the sequence at the end of that gene. The mRNA can be translated without further modification. But in eukaryotes, do you remember about RNA processing? I told you that there needs to be some RNA processing. So in eukaryotes, RNA polymerase II transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. So what does that mean? That sounds super confusing because we haven't talked about it yet, but we're going to talk about that next. Next, we're going to talk about RNA processing, right? So this doesn't seem very straightforward because we haven't talked about RNA processing. And let me ask you this. Can you beat Wicket? Does RNA processing occur in prokaryotes, including bacteria? No, that's right, Wicket. RNA processing does not occur in bacteria. Remember, transcription and translation occur at the same time. The mRNA is ready to go. It's ready for translation in a bacteria, in a prokaryote. Whereas in a eukaryote, remember, just because we've done transcription, initiation of transcription, elongation of transcription, and termination of transcription, it doesn't mean we're ready for translation. It doesn't mean we're good to go. Remember, at this point, we have our pre-mRNA. And the pre-mRNA needs to be modified. Remember, I told you there are 3 modifications. We need the 5' modification, the 3' modification, and then we need to modify some stuff in the middle of the mRNA as well to make our mature mRNA. So we are not ready for translation yet in eukaryotes. So next, let's talk about what in the world does RNA processing mean? What are these 3 modifications that need to occur on the eukaryotic mRNA? Let's talk about that next. Now remember, there are 3 modifications that occur during RNA processing. But first, I'm just going to touch on the ones that happen at the ends of the mRNA, the 5' end and the 3' end. Let's just talk about those 2 modifications first. At the 5' end of the mRNA, this receives a modified nucleotide 5' cap, and the modification is called the 5' cap. I'm going to show you what that looks like in a second. On the 3' end of the mRNA, it gets what's known as a poly-A tail, a polyadenosine tail. So we got to look at that in a minute too. Take a look at the mRNA. Here's your mRNA, your pre-mRNA. Okay, let's focus in on the 5' end first. Do you see this 5' end? The 5' end of your mRNA gets a cap. And you might be wondering, what is the cap? This is the cap right here. See, it's called the 5' cap. And all it is a guanine. Do you guys remember the nucleotide G for guanine? It's a purine, so it has 2 rings. This is guanine. There's your sugar, your pentose sugar, ribose, and you have a triphosphate. So this is guanine triphosphate, and in addition, it has a small methyl group. Okay, so it's called methylated guanine. And what's really interesting is that a guanine, a methyl-guanine, is added to the 5' end of the mRNA, but it's added in reverse. So what do I mean? What's normally at the 5' end of mRNA? What's normally at the 5' end of a strand of nucleic acid? Can you beat Wicket? That's right, Wicket. There's usually a phosphate group at the 5' end of a strand of DNA or RNA. So there should be a phosphate group here. Now, what's strange is that this methyl-guanine is added 5' to 5'. What that means is these phosphates, these 5' phosphates on this methyl-guanine are connected to the 5' phosphate on the mRNA. Isn't that weird? So it's like a reverse joining. The 5' end of methylguanine is connected to the 5' end of the mRNA. So it's almost like they added this nucleotide, this methylated nucleotide, but they add it backwards, okay? Isn't that neat? So that's called a 5' cap. And a 5' cap is simply when you add a methylguanine 5' to 5' and you connect it to the 5' end of the mRNA. And you might be wondering, why do we do this? Why do we add that cap on the mRNA? Well, it seems to facilitate the export of mRNA to the cytoplasm. Remember, mRNA needs to leave the nucleus to meet up with the ribosomes in the cytoplasm. It helps protect the mRNA from hydrolytic enzymes, so it helps keep the mRNA from breaking down. And it helps ribosomes attach to the 5' end. It helps the ribosomes to recognize the mRNA, okay? Now, let's talk about the 3' end. Now, look at the 3' end's a little bit more interesting. Here, look at this. At the 3' end, which ended here, by the way, this yellow color, the mRNA ended at this little line right here, at this red line. Now, at the 3' end of eukaryotic mRNA, there's a sequence AAUAAA, and this is known as the polyadenylation sequence. What in the world does that mean? That means that shortly after this sequence, transcription is finished. So the pre-mRNA only was this long at the 3' end right here. It ended right here. An enzyme called polyadenylase comes along afterwards, and this is known as post-transcriptional modification. This means that it occurs after transcription. An enzyme comes along and it recognizes this adenylation site, AAUAAA, and when it sees that, it joins and it adds a bunch of A's. These are the same A's as RNA, you know, A's, G's, C's, and U's, right? These are A's. So it's called a poly-A-tear or polyadenylation. You put a number of A's, and by a number, I mean a number. There could be hundreds of A's added to the 3' end of the mRNA tail. So, and again, why in the world does the cell add a bunch of A's to the end of the mRNA post-transcriptionally? Again, it helps protect the mRNA from breaking down, and it helps with the export of mRNA to the cytoplasm. So does that make sense? Again, just to summarize, here's a different image of the same thing. You have a 5' cap where a methyl-guanine is added 5' to 5' to the 5' end of the mRNA. And you have a poly-A tail where you have 50 to 250 adenine nucleotides added to the 3' end of the mRNA post-transcriptionally shortly after the polyadenylation signal on the mRNA. Isn't that neat? So these are two modifications, but remember, there's still a third modification, and this one takes even more paying attention to understand correctly. So let's focus in on what's the third and final modification on the mRNA of eukaryotes. The third and final modification that needs to occur during RNA processing is known as splicing out of intron sequences. So to understand this, we need to understand that eukaryotic genes, they are unlike prokaryotic genes in that they have these non-coding regions of the gene called introns, and they have coding regions of the gene called exons. And until we understand what that means, we can't fully understand the third modification. So let's go to the board real quick where I'm going to try to explain to you what introns and exons are, and then also how this third modification works. So let's go to the board real quick. All right, so here's something you need to understand about eukaryotic genes. Let's say this is a gene. This is the portion of a chromosome that codes for a particular protein. So for example, if this was the insulin gene, it would code for the insulin protein. Or if this was the hemoglobin gene, it would code for the hemoglobin protein. So what you need to understand is that in eukaryotes, and not prokaryotes, in eukaryotes there are parts of the gene that actually code for protein, and then you have parts of the gene, which I drew here in red, that don't. They're filler. They used to call it junk DNA, but now we know it has other functions besides coding for protein, but it's not parts of the gene that actually code for protein. So the parts that do code for protein are called exons. So this would be exon, EXON 1. This one would be the next exon, EXON 2. And then you have exon 3. Exon 1, 2, and 3. So this gene in particular has 3 parts to it that code for this particular gene. But then you also have the other filler parts, and the fillers are called introns. So this would be called intron, I-N-T-R, intron, O-N, intron 1, intron 2, and intron 3. Intron 1, 2, and 3. So why do we care? Again, these introns, this filler information in the middle of the gene is not present in prokaryotes. It's only found in eukaryotes. In a prokaryote, the entire gene is like one big exon because everything codes for a protein. In a eukaryote, you have introns. Here's the problem. When the RNA polymerase attaches to the DNA and copies the DNA into mRNA, right? Remember mRNA? What are you copying? When you copy the DNA into mRNA, that pen does not want to stay, you copy all of it, don't you? You copy all of that gene from beginning to end, don't you? Which means not only did you copy the exon information, you also copied the filler information, the intron information. Hey, no, Tig wants in. Come on in, Tig. Yeah, come on in. We're making a video. Now he's sitting outside yelling. Come on in. Yeah, come on in. Okay, or just stay outside and yell. He does that. So anyway, this in a eukaryote, this would be called what? Is it mRNA? No, remember it's called pre-mRNA. Pre-mRNA. So the pre-mRNA, if you recall, this would be the 5' end of the pre-mRNA. This here would be the 3' end of the pre-mRNA. The 5' end gets a cap, right? Cap. A 5' cap. The 3' end gets a poly A tail, right? You get a bunch of A's added on. Remember that? And now for the third and final modification on your pre-mRNA. You see that red information, the intron information? Remember I said that information is really not useful for the ribosome to make a protein. It's not useful information. So we actually have to cut that information. Now imagine if I had scissors, molecular scissors, and I could just cut out the red parts, the intron parts. Because remember, the blue part is exon 1, exon 2, exon 3, right? But you have the red parts, which are intron 1. Oops. I'll just call it I1, I2, and I3. We have to remove the introns. This is called splicing out the introns. A special enzyme called the spliceosome comes along and it literally removes, it splices out, excises the introns. Okay, you need to know that the introns are excised by the spliceosome. The spliceosome is the enzyme that will splice out or excise the intron. So what are you left with? You're going to be left with, if you cut out all the introns, you're going to be left with a shorter, actually even shorter, a shorter mRNA. This short mRNA is called your mRNA, mature mRNA. Here's the 5' with a cap. Here is the 3' with a poly A tail. And it's a much shorter mature mRNA because we've cut the intron information out. Okay, so those 3 modifications, you don't need to know in great detail, but you need to know that in eukaryotes, you have intron information, and when the mRNA is produced, that mRNA is quite long because it includes that intron information. So the 5' end of the mRNA needs a cap. That's called the 5' cap. The 3' end gets a poly A tail. And the third and final step, excision of the introns, right? Removal of the introns by the spliceosome. In the next step, I'll show you what the spliceosome looks like. So again, to summarize real quick, look at here. If this is the eukaryotic gene across the top, the light blue areas are the coding exons that code for proteins, and the purple areas are the introns that do not code for proteins. However, remember, when you transcribe this gene, this transcriptional unit, you're copying both the useful exon information as well as the non-useful intron information. After transcription, you have a long pre-mRNA, which includes these gold portions that are your exons, as well as these purple portions, which are the non-coding introns. Remember that the 5' end gets the Methyl-Guanine 5' cap. The 3' end gets the polyadenylation, the poly A tail. Then at that point, what do we need to do? Can you beat Wicket? That's right, Wicket. We need to splice out the introns. So these purple parts need to go away. And when we splice out those introns with the spliceosome, the mRNA is now shorter, right? Because we cut out the purple portions, the introns. Now you've got your 5' cap, you've got your 3' poly A tail, and you've removed the introns. This means exon 1 is right next to exon 2, right next to exon 3, right next to exon 4, and your mature mRNA is going to be shorter, and it's ready to export to the cytoplasm to meet up with ribosomes for translation. Here it is again in your textbook, your current textbook, 5' cap, 3' gets a poly A tail. You've got your intron. This is a really long intron. Actually, you've got a short intron here, a long intron there. The introns get spliced out, the exons get spliced together, and you have your short mRNA that's mature, ready to be exported to the cytoplasm and for translation. And remember, again, can you beat Wicket? What enzymes are responsible for splicing out the introns? That's right, Wicket, the spliceosome. So let's take a look at the spliceosome. With the spliceosome, these are responsible for removal of the introns. The spliceosomes consist of a variety of proteins and several small RNAs that recognize the splice sites. The RNA of the spliceosome also catalyze the splicing reaction. So take a look here. If this is the 5' cap, and over here you have your 3' poly A tail somewhere on the right. Let's say dark red. Red is an exon. So you've got exon 1 in red. Then in peach color, you have your intron. And then in red, again, you have exon 2. Remember, we need to remove this intron, and we need to join exons 1 and 2. And again, the spliceosome, which is made up of protein and small RNAs, the small RNAs are depicted here in red. The spliceosome will bring the exons together and literally loop out, splice out, or cut out the intron. This is known as cut out intron or spliced out intron. And now the result is that you have exon 1 joined directly to exon 2. So this is how pre-mRNA is processed into mature mRNA by removal of the non-coding intron regions. Now, while we're still on the topic of eukaryotic RNA splicing or intron splicing, eukaryotic genes can sometimes encode more than one kind of polypeptide. This means that some genes give you more than one protein result. And this depends on which segments are treated as exons during splicing. This phenomenon is called alternative RNA splicing. So let me show you at the board something really neat that eukaryotes can do, because guess what? There are somewhere upwards of 80,000 different proteins that your cells can make. However, there's only 25,000 genes. So somewhere the math doesn't add up, does it? Think about that. If a gene is supposed to code for a protein and you only have 25,000 genes, how many different proteins should you be able to make? Right, Wicket, 25,000. If a gene codes for a protein, well then you should have 25,000 proteins because you have 25,000 genes. However, what if I were to tell you that your cells could make upwards of 80,000 different kinds of proteins? That wouldn't make sense, right? So what explains the fact that you can make so many different types of proteins from a limited number of genes? The answer is called alternative RNA splicing. And of course, this is only applicable to eukaryotes because only eukaryotes do RNA splicing, right? And what is RNA splicing, alternative RNA splicing? It's a way for us to make more than one polypeptide. This means more than one protein from the same gene by alternating the way the genes are spliced. The mRNA is spliced. Now, let me take you to the board for a proper explanation so it all comes together. All right, so here I'm going to explain alternative splicing. It's a process only found in eukaryotes, and it takes advantage of the fact that eukaryotes have exons, multiple exons. Like, remember this example I showed you earlier, exon 1, exon 2, exon 3 for this gene. Remember what I said? The entire gene is copied, so you get a pre-mRNA that's quite long. It includes all the exons and all the intron information. Splice out the introns to give you a mature mRNA that contains information for exon 1 next to exon 2, right next to exon 3. So all the exons are now right next to each other, ready for translation, right? So what's alternative splicing? Alternative splicing is, again, only found in eukaryotes. It is a process by which eukaryotes can make more than one protein, right? More than one gene product from the same gene. How can you possibly do that? Well, what if you shuffled the exons around or left an exon out or added an exon? Let me tell you. So for example, if the ribosome were to come along and translate this, right? Translation. If the ribosome were to translate this, it would translate exon 1, exon 2, and exon 3 information into a polypeptide protein, right? Into a protein. But what if I did an alternative to this splice, an alternate splice? So for example, what if I, during splicing, you know how I spliced out my introns? What if I also, for example, spliced out exon 2? Did you see that? What if I also spliced out at exon 2? Would that not result in a much shorter mRNA with exon 1 right next to exon 3? Because we've left out exon 2 information. So now you have a really short mRNA that has different information. It's got exon 1 and exon 3. You've left out the exon 2 information. Now, when the ribosome makes that protein, is it going to make the exact same protein? No. This protein is going to be translated differently. It's going to be a shorter protein, and it's going to have a different structure to it, right? So in this case, the same gene gave you two different proteins due to alternative splicing. All right, let's go back to our overview slide here and see what we've covered so far. Remember, gene expression is the process by which cells produce proteins from genetic information, the DNA. And this is a two-step process, starting with transcription, where the gene is copied to complementary mRNA. And remember, there's a difference between how this occurs in prokaryotes versus eukaryotes. In prokaryotes, transcription occurs, and before transcription's even finished, translation can begin. So transcription and translation occur concurrently in the same place and the same time, that place being the cytoplasm. And then we talked a little bit more in depth about the eukaryotic gene expression, starting with transcription. Remember, the gene is transcribed into a long pre-mRNA. And now we know why the pre-mRNA is so long, right? It's because it includes both exon information, which codes for protein, as well as intron information that does not code for protein. And then do you remember we just talked about RNA processing, by which the pre-mRNA obtains a 5' methyl-guanine cap, a 3' poly A tail, and the introns are spliced out by our friends, the spliceosome. Okay, that's where we left off. But remember, now it's time for translation. We haven't made a protein yet. We need to do translation, right? And translation occurs in the cytoplasm with the ribosomes. So the ribosomes are going to be our star player now. The ribosomes are responsible for translation. The ribosomes can read the message on the mRNA, where they can read that genetic message to string together the proper amino acids in the proper order to form the proper polypeptide, making the protein. So before we delve into translation in detail and learn how translation works, why don't we take a quick break time with Gizmo and Wicket and see what these two little guys are up to, and then we'll be right back. All right, welcome back from a very intense break time with Gizmo and Wicket. So again, where we left off, the mRNA is ready for translation. And how does this occur? First of all, the mRNA needs to meet up with the ribosome, right? So let's talk about what happens during what's called initiation of translation. How does translation begin? Now, one thing you need to understand is that translation doesn't begin at the first nucleotide on the mRNA. It occurs downstream a bit. What happens is the small subunit of the ribosome. Remember how the ribosome looks like a hamburger bun with a large subunit and a small subunit? Well, the very, very first thing that happens is the small subunit of the ribosome. It finds the 5' end of the mRNA, and it's searching for what's known as the Ribosome Binding Site on mRNA. And as its name suggests, this will allow the ribosome to bind. And now the ribosome searches for a particular code on the mRNA. Now we're looking at the mRNA codes. And so to do that, we need to understand what the mRNA codes mean. So let's go over to what's called the Codon Table so that we can understand the genetic code and just how the genetic code codes for proteins. Let's do that next. Let me talk to you about the genetic code for a minute. I want to tell you about the language of DNA, the language of genetics. But first, as a comparison, I want to ask you if you've ever seen a dictionary, like an English dictionary. I don't have one on hand, but if you've ever seen a modern dictionary, it's even thicker than your textbook, your Campbell's textbook here. A modern English dictionary can be twice as thick as this, right? I mean, if you think of the Merriam-Webster dictionary, for instance, there are so many words in there, and every year they're adding more and more words. It's getting out of hand how many words there are. But the reason I bring that up is because English is, amazing language, and we have a lot of words and ways to express ourselves, but you would think that the genetic code would be even more complicated with more words, right? Because think of all the beauty in life and all the different creatures, everything from viruses, which are not creatures, by the way, viruses to cells, bacterial cells, archaeal cells, photosynthesizers, plants, animals. Think of all the different types of species out there. And, the DNA codes for all this diversity of life. So you would think that the genetic code should be really big and cumbersome and convoluted and, much bigger than a dictionary, right? But it turns out that is actually the opposite. The genetic code is surprisingly simple. Well, let me tell you some differences between the English dictionary and the genetic dictionary. English dictionary includes words that are one letter long, like A, right? Or I, A, these types of one letter long words. There are 2 letter long words, 3 letter long words, 4 letter long words, and some words are, 20 letters long, right? Now, one key difference between English and the genetic language is that every word in the genetic code, every word in the genetic language is only 3 letters long. And you know what those letters are from before, right? Those letters are called nucleotides. Remember, there are A's, G's, C's, and T's. That's it. That's the whole alphabet. Whereas in English, we have 26 letters, and those letters are used to make words that are 1 letter long all the way up to more than 20 letters long. In the genetic language, every single word is only 3 letters long. And these 3-letter long words are called codons. So for example, AAC is a word. It's a codon. It's called the Triplet Code. CCG is a codon. ATG is a codon. TTC is a codon. CCA is a codon. You see, so every word is only 3 letters long, but the alphabet has only 4 letters, not 26 like the English alphabet. So as I mentioned earlier, you could fit the entire genetic dictionary, every possible genetic word, you could fit it on a postcard. I kid you not. Let me show you the postcard. Let me show you the entire dictionary in the genetic language, the genetic code. You ready for this? Let's pull it up. This right here in front of you is every word in the genetic language, all summarized on a single table that could fit on a postcard. This is the key to all life, all the different organisms, no matter how simple, no matter how complex. They are coded for by this series of 64 codons. Yes, there are 64 possible codons, and they are listed on this table. This is known as the Codon Table. This Codon Table is actually an mRNA codon table, so instead of T's, you see U's. But, don't get confused. Those U's were copied from T's, right, on the gene. So, don't get confused. Where you see a U, that corresponds to a T, right? But we're talking about reading the mRNA. If you're reading the mRNA, these are the words. These are the codons. And this is the genetic code. Okay. Now, what the ribosome is going to do is that it's going to read these codons on the mRNA. And don't forget, the ribosome does not begin reading at the 5' end of the mRNA, does it? Instead, it scans for that ribosomal binding site, and then it's looking for a particular code. Look closely at this codon table and just analyze it for a second. Maybe pause the video and tell me what you think based on just looking at this table. Tell me what you think the ribosome would be looking for. Which codon on this table would the ribosome be interested in finding first, do you think? Right, Wicket chimed in with the answer. You see this codon right here? This codon, AUG, or on DNA, it would be ATG. This codon is special. This codon is known as the start codon, and it tells the ribosome where to start translation. The ribosome will not start translating the message until it finds the first start codon, the first AUG that's closest to the 5' end of the mRNA. Okay, so that's known as the start codon, and that start codon tells the ribosome to start translation. And conversely, let me show you something else. There are 3 stop codons, UAA, UAG, and UGA. These are the 3 stop codons. And these 3 stop codons, you don't need all 3. Any one of these 3 will tell the ribosome to stop translating the mRNA. Okay? And by the way, again, this stop codons appear before the 3' end, before the very far 3' end of the mRNA. So there are regions that are not translated at the 5' end of the mRNA, and there are regions that are not translated at the 3' end of the mRNA. Take a look. At the 5' end of the mRNA, there's a region that is not translated, and that is known as the 5' untranslated region, or UTR. This untranslated region is the region before the start codon. Can you beat Wicket? What was the start codon? That's right, as always, Wicket. AUG. So the ribosome will attach somewhere near the 5' end of the mRNA at the ribosomal binding site. It will then search for that AUG start codon, and only then will it start translating the rest of the message into protein. The series of nucleotides it skips to get to the first codon, the start codon, is known as the 5' untranslated region, because this region of the mRNA is not translated. Then, between the start codon and the stop codon, the message is translated into protein. At the stop codon, remember the 3 stop codons, UAA, UAG, UGA. These are the stop codons, and those tell the ribosome to stop translating. Anything after the stop codon is not translated, including the poly A tail. So this is known as the 3' untranslated region. Again, just because the mRNA is this long doesn't mean all of it codes for protein. The protein coding part starts at the start codon and ends with the stop codon. The rest of the mRNA molecule is not translated. So now what do we know? We know that the entire dictionary of the genetic language can fit on this postcard. There are 64 codons total. We just discussed 4 of them, did we not? AUG, the start codon, UAA, UAG, and UGA, which are the stop codons. But what about the rest of the codons? Let's talk about these codons. What do the codons code for? What are these words? Well, these words, these codons, code for amino acids. Different codons code for different amino acids. All the ribosome does is read the codon on the mRNA and pair it up with its corresponding amino acid. So for instance, look at this. CUU, the codon CUU, translates to the amino acid leucine. LEU is its 3-letter code for leucine. The letter L also represents the 1-letter code for leucine. And different codons code for different amino acids. Do you all remember that there are 20 different naturally occurring amino acids, 20 amino acids that make up your proteins? Remember we discussed proteins? Proteins are just a long chain of amino acids, and you have the primary structure of the protein, which is just the chain of amino acids in order, and then those structures fold into secondary structures like alpha helices and beta sheets. And then those fold further, thanks to R group interactions, into tertiary structures, right? You remember all that? So that's how a protein looks like, right? That's how a protein forms and folds. But the protein is made up of 20 amino acid. How many different codons are there? Can you beat Wicket? That's right, Wicket. There are 64 codons, but only 20 amino acids. Do you see a problem here? It's like having 64 words, but only 20 meanings. Does that kind of make you scratch your head? Why are there only 20 meanings if there are 64 words? Well, take a look and see. Did you know that you can have more than one word, more than one codon coding for the same amino acid? So for instance, look at leucine, CUU, CUC, CUA, and CUG. All of these codons code for leucine, the amino acid. So there can be more than one codon coding for the same exact amino acid. And that makes sense because otherwise, what are you doing with 64 words and 20 meanings? There must be some redundancy there. And so they consider the genetic code as redundant. What does that mean? There are more than one codon which may specify a particular amino acid. Okay, so the genetic code is redundant. Do you notice how there are multiple codons that code for the same amino acid? For instance, again, GGU, GGC, GGA, GGG, all of these code for the amino acid glycine. Okay, so again, the genetic code is redundant. However, I need you to understand something very important, and that is that the genetic code is not ambiguous. Okay, you see here, but the genetic code is not ambiguous, which means what? It means no codon specifies more than one amino acid. So although the genetic code is redundant, meaning more than one codon can code for a particular amino acid, the genetic code is not ambiguous. So for example, CCC codes for proline, but it does not code for anything other than proline. So make no mistake, although there's more than one codon that codes for proline, these codons do not code for anything other than proline. That's why you need to remember that although the genetic code is redundant, the genetic code is not ambiguous, and that's what that means. Okay? Now, again, these codons code for amino acids. And even the start codon, remember the start codon, AUG, tells the ribosome to start translation, but it also codes for an amino acid. AUG codes for the amino acid methionine, MET, or M for short, methionine. So if this is true, let me ask you something. If this is true, doesn't every protein begin with methionine? If the start codon itself codes for methionine, doesn't that mean that every protein will start with methionine? Can you beat Wicket? That's right. Exactly, Wicket. Yes, every protein would start with methionine. Why? Because the start codon itself tells the ribosome to place methionine as the very first of the amino acids in the sequence, in the polypeptide chain. Isn't that interesting? But conversely, let me show you the stop codons. You guys remember we talked about these stop codons. UAA, UAG, UGA. Do any of the stop codons code for an amino acid? No, right? So the stop codons do not code for an amino acid. They simply tell the ribosome to stop stringing amino acids together, to stop creating a protein. Okay, so take a look. The ribosome is searching for the start codon. It will place methionine at that location. The ribosome will then read every 3-letter word, every codon from that point on, stringing the appropriate amino acids together with peptide bonds, forming a polypeptide chain. And it will do so until it encounters what? Until it encounters any one of these 3 stop codons, at which point it knows it's completed its task. It's strung enough amino acids together. And most proteins are pretty long, actually. Most proteins are, they could be hundreds of amino acids long, right? So you start with the start codon. You then read every 3 letters, stringing the correct amino acids together. And then eventually, after a few hundred amino acids, the ribosome will encounter a stop codon, and it will stop making a protein. Isn't that neat? And remember, this process occurs concurrently in prokaryotes like bacteria. Check it out. Here, in this blue line represents a gene. And these little guitar pick looking things, as you know, these are your RNA polymerase friends. RNA polymerase proceeds along the gene, forming the RNA transcript in red. This in red is the mRNA transcript. And do you remember what I said earlier? Before transcription is even complete, ribosomes can chomp down on the mRNA and start translating that message into proteins. So here you have, again, these little white structures are the RNA polymerase. They are transcribing the DNA into mRNA, which is in red. Immediately, the ribosomes, which look like these hamburger buns, can attach and start translation. And they're forming the polypeptide chain. They're forming proteins. The proteins are in purple. Here you can see that the ribosomes are reading the mRNA message from 5' to 3'. And every codon, starting with the first codon, let me see if you can beat Wicket. What do you think this pearl is right here? This amino acid right here, which amino acid of the 20 do you think it is? Right, Wicket. It is methionine, M-E-T, methionine. Why? Because the start codon codes for methionine. So I can tell you, without even knowing what protein this is, I could tell you this amino acid right here is methionine. And then next to it would be whatever amino acid corresponds to that codon. And next to it is whatever amino acid responds to the next codon. And so the ribosome just proceeds codon by codon, reading every 3 letters on the mRNA and putting down the correct amino acid. Isn't that neat? And like I said, look at this. Translation can begin before transcription is even complete, and this is known as coupled transcription and translation in bacteria. These are known as polyribosomes because several ribosomes chomp down on the same mRNA, and so you can see you can make a lot of protein product in a short amount of time with gene expression in bacteria. So now let's go into a little more detail about how translation works. We can look at it more closely. Exactly how does the ribosome make the protein? Well, here you can see, remember the ribosome is this organelle that has a large subunit, which I call the large hamburger bun. And then down here you have the small subunit, the small hamburger bun. And these 2 subunits can come apart and they can come together, right? And so this, I want you to think of the ribosome as a printer. You know, just like the printer at your house, you have a computer printer, you know, or a 3D printer, you know, a printer, right? Now, let me ask you something. Do you think your printer is useful if you have absolutely no printer cartridges? So think about it. If you don't have ink cartridges or printer cartridges, your printer is pretty much a paperweight, right? It's pretty useless. Same thing with even a 3D printer, right? If you have no filament, cartridges or no filament, well, then your 3D printer is useless, right? So printers are pretty useless without toner, without cartridges. Does that make sense? So think of the ribosome as a 3D printer of proteins, right? And you know what a protein is, right? A protein is a bunch of amino acids linked together with peptide bonds, right? Remember, a protein is simply a long chain of amino acids, and then that long chain folds into that tertiary structure. But the ribosome is simply a 3D printer that links the correct amino acids together in the correct order by using the mRNA message, right? Using mRNA as a guide, okay? So you can think of the mRNA as the information that the printer is using to print the correct protein, and the ribosome itself is the printer. So doesn't that leave one more thing to explain? What is that thing, do you think? Can you beat Wicket? That's right, as always, Wicket. The printer cartridges, right? What are the printer cartridges that the ribosome uses? So next, let's talk about the printer cartridges. And I just want to turn your attention to these 3 little cavities inside of the ribosome. There's a cavity called E, there's a cavity called P, and there's a cavity called A. These are the cavities in which the printer cartridges fit. So printer cartridges fit in these 3 cavities. So I want you to think of these 3 cavities as somewhere that the printer cartridges for the ribosome fit. So now let's talk about what these printer cartridges look like. Let's talk about that next. Okay, here are the printer cartridges. This is it right here. This is a cartridge that fits into one of those 3 cavities we just talked about. You can think of this structure right here in kind of aqua. This structure is the printer cartridge for the ribosome. It's actually called tRNA. It's made of RNA, but it's called tRNA for transfer RNA. And this structure, this is the cartoon, but what this cartoon represents is this RNA structure here. This is what it is. And remember, RNA is usually single-stranded, and this printer cartridge is a single-stranded RNA. It has a 5' end that loop-de-loops around, and then here's its 3' end. So it kind of binds to itself at these spots. So it forms like an upside-down L structure. Here is another look at the tRNA. The tRNA is drawn here as a flat clover structure. Here you can see it as a more three-dimensional structure, and on the right is the cartoon diagram of the tRNA structure. So these small pieces of RNA that look like upside down L's, these small pieces of RNA are the printer cartridges for the ribosome, okay? You can think of these as printer cartridges for the ribosome. So next, let's talk about why it's a printer cartridge, okay? So there are two important parts to this printer cartridge. There's this end down here. You see this end down here, this end down here. There's 3 nucleotides on this RNA, on this tRNA, 3 nucleotides that are so important because these 3 nucleotides interact with the mRNA. These 3 nucleotides are called the anticodon. You see, so for example, the anticodon on this tRNA is AAG. That's the anticodon. Now, if that's anticodon and this AAG anticodon is going to interact with and bind to mRNA, what codon would you say that this anticodon would bind to? Think about it logically. Can you beat Wicket? Which codon on the mRNA would the anticodon complement AAG bind to? Think about it for a second and let me see if you can beat Wicket. That's right, Wicket. So the codon that would bind the anticodon AAG would be UUC. It would be UUC. So the codon on the mRNA would have to read UUC for this tRNA, whose anticodon is AAG, to bind to it, right? Due to complementary base pairing. Remember, A's pair with U's, G's pair with C's. So the codon that this tRNA could bind to would be UUC. And by the way, yes, there are many different tRNA, and these tRNA have different anticodon. And those anticodon recognize what? They recognize codons, right? So remember, the codon that was recognized by that tRNA was UUC. So let's see what codon that would correspond with. Can we find UUC? Here it is. On the codon table, the codon UUC, which would bind to the anticodon that I just showed you, AAG, corresponds to the amino acid phenylalanine, FHE, or F for short. Phenylalanine is the amino acid that is coded for by UUC. Okay, keep that in mind. Okay, jumping back to this slide, take a look. Here you can see that this is a tRNA with the anticodon AAG, which we just confirmed should bind to the codon UUC for phenylalanine. Now look at the other end. This is the other important end here. This is called the Amino Acid Attachment Site. These tRNA get loaded up. They get loaded just like your printer cartridge is loaded with ink. These tRNA molecules get loaded with one amino acid, and that amino acid gets joined to this amino acid attachment site right here on the 3' end of this tRNA. There's an amino acid that gets attached here. But let me ask you this, and let me see if you can beat Wicket. Which amino acid do you think would be preloaded on this printer cartridge, the printer cartridge with the anticodon AAG? It has to be one of the 20 amino acids that this tRNA is preloaded with. Which one would you say that would have to be according to your codon table? That's right, Wicket. Phenylalanine. Guess what? If the tRNA is the one for AAG, that means it recognizes UUC. That means that this tRNA has to be preloaded with the amino acid phenylalanine. And how do these tRNA get preloaded? Well, you don't need to know this in detail for my class, but there are these enzymes called Aminoacyl-tRNA synthetases, and these enzymes load tRNA with the appropriate amino acid. So here you can see, for example, look, here is a tRNA. This is called an uncharged tRNA. You know why? Because it does not have an amino acid attached, right? This tRNA is uncharged until it's recognized by a aminoacyl synthetase, which will grab the tRNA. It will also grab the appropriate amino acid. In this case, this tRNA is the one with the anticodon AUA, which is complementary. It's anticodon for the codon for tyrosine. The amino acid is tyrosine. So this synthetase will grab the tRNA. It will grab the appropriate amino acid for that tRNA, and then it will use the power of ATP to charge that tRNA. So here you can see that the tRNA is charged. Again, if this tRNA has the anticodon AUA, it's going to be loaded with the correct amino acid for the codon UAU. Does that make sense? Which happens to be tyrosine in this case. Okay, but which one were we just talking about? We were just talking about this guy, this anticodon AAG, right? AAG. So if this tRNA was the one with the anticodon AAG, then this synthetase would have put the amino acid phenylalanine on it, right? So it would have had phenylalanine attached. Does that make sense? And you can think of these tRNA that have an amino acid attached, the correct amino acid attached, these tRNA are known as charged tRNA. The tRNA that do not have the appropriate amino acid attached are called uncharged tRNA. And what happens in the background is all of the correct tRNAs are matched up with their appropriate amino acids. So the ribosome has access to thousands and thousands and thousands of these tRNAs that are charged. Like, it's almost like, imagine if your printer was surrounded with loaded ink cartridges and it just needs to grab the correct one when it needs it. Think of it that way. All right, so here we are back at initiation of translation. Remember what happened. The small subunit, the small hamburger bun of the ribosome, identified the 5' end on the mRNA, and it found it by finding the ribosome binding site. Next, which codon is it going to search for? It's not going to start translating right away, is it? Which codon do you think the ribosome is going to search for? And when it does, the ribosome slides through the message. The ribosome moves from left to right. It reads through the mRNA, and it's searching for a particular codon. What codon do you think that is? Can you beat Wicket? That's right, Wicket. The start codon. We need to find the first start codon. And do you remember the start codon reads AUG? That's the start codon. And the start codon, remember, it also codes for the amino acid methionine, or M. Now, let's go back to our diagram here. So again, the small subunit of the ribosome binds to the 5' end of the mRNA at the ribosome binding site. It then scans and looks for the first start codon. So let's see that happen. Here you go. At this point, the small subunit of the ribosome has found the start codon on the mRNA. How do I know this is the start codon? Well, on the mRNA, it should say what? It should say AUG. Does it say AUG? Let's zoom in and take a look. That's right, it says AUG, which means what binds to that? This is the tRNA that recognizes AUG. So what anticodon does this tRNA have to have if it's going to pair up in a complementary fashion with AUG? It should have UAC. C, right? So guess what? Only the tRNA with the anticodon UAC is going to recognize the start codon AUG, and only this tRNA is going to bind. And which amino acid do you think it brought with it? Which amino acid was it preloaded with from that synthetase enzyme we just talked about? It's preloaded with methionine, right? This is the amino acid methionine, which is bound to that 3' end of this tRNA molecule because its anticodon reads UAC, and that binds to AUG. I hope this makes sense. Again, the small subunit of the ribosome finds the start codon. It then finds the initiator tRNA, or I call it the start tRNA, which brings along with it methionine. Doesn't that make sense? Isn't that neat? And at this point, energy is used, GTP is used, and boom, the large subunit, the large hamburger bun attaches at this point, and now you have initiated translation. This is called Transcription Initiation Complex. This is the very first step to translation, and here is the first amino acid in your protein chain, in your polypeptide chain. This is what needs to be done. So now you might be asking, what's next? Well, next is called elongation of translation, because we need to read the message, right? Which way do you think we're going to read? Do you think we're going to read towards the 5' end of the mRNA or towards the 3' end of the mRNA? We're going to read towards the 3' end, right? Because remember, we just bound to the 5' end, and then we skipped some nucleotides and we found the start codon. We need to read towards the 3' end of the mRNA. So by the way, look at this tRNA. Which site did this first tRNA bind to? It bound to the P site. Remember, this was the E site. This is the P site. This is the A site. I'll tell you what these sites do in a minute. But at this point, this tRNA is in the P location, in the P cavity. You can think of it that way. The next thing that will happen is, guess what? A tRNA is going to enter the A site. You can think of the A site as the acceptor site. The A site accepts the next fresh printer cartridge. It accepts the next charged tRNA. But let me ask you this. Do you think just any tRNA is going to come into the A site? Do you think just any random tRNA is going to enter the A site? What do you think dictates the identity of the tRNA that's going to enter the A site? Can you beat Wicket? That's right, as always. So yes, only the tRNA that corresponds to this codon. See these 3 letters? Whatever these 3 letters are is the codon. You see? 1, 2, 3. If this codon says C, 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 then the tRNA that corresponds to that, G, 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 is the only one that's going to come in and bind. And it's going to bring with it the corresponding appropriate amino acid. Does that make sense? So again, if this says C, 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 only the tRNA with the anticodon G, G, G would come in and bind. And let me quiz you. Which amino acid would it bring along? If the codon is C, 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 and the anticodon is G, 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 which amino acid should that tRNA bring with it? Can you beat Wicket? That's right. Proline. Check it out. Proline's codon is C, C, C. C, C, C corresponds to proline. So guess what? If, I'm just making up an example, if this next codon next to the start codon, AUG, if this says C, 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 then the tRNA with the anticodon G, G, G will come in and bind, and it'll bring with it the amino acid proline. Does that make sense? That's what's going to happen next, and that is the next step. All right, so where did we leave off? Check this out. Here you can see the ribosome. This is the 5' end, right? And the ribosome found the start codon. Let's pretend this here is the start codon AUG. And then remember I said the next tRNA enters, the next charged tRNA at that enters at the A site. Do you remember that? So you've got this going on. So let's take a close look here. Take a look with me. Here you have the start codon AUG, which remember binds to the start tRNA or the initiator tRNA, which has the anticodon UAC, and it would be bound to which amino acid? Can you beat Wicket? That's right, methionine, M-E-T, methionine. And next, remember what I said next? Next, the codon, let's pretend the next codon is CCC, which means the charged tRNA with the anticodon GGG would bind, and which amino acid would it bring with it? That's right, proline. So now you have methionine here attached to the tRNA in the P site. You have proline here attached to the tRNA in the A site. And now here is where the magic happens. Okay, so again, I told you that the A site is for accepting fresh tRNA. Well, what's the P site for? The P site is called the site for Peptidyl-Transferase Activity. That's a mouthful, but what that means is it has the ability to make the peptide bonds. What is a peptide bond? Do you remember? It's the bond between amino acids. Just because I have an amino acid here and I have an amino acid next to it, is that a protein? Or do I have to link these two amino acids together? Think about that. Don't I have to link methionine to proline? Don't I have to link this amino acid to that amino acid? So imagine if I have scissors and I cut the amino acid methionine away from this initiator tRNA. So I hydrolyze this bond, right? I hydrolyze this bond right here. And then I make a bond between this carboxyl group and the amino group on the proline. So that's what the P site does. The P site makes the peptide bonds. If for simplicity, you can think of it as the P site makes the peptide bonds. The P site has the Peptidyl-Transferase activity. The P site cuts the amino acid off of this tRNA and transfers it to the next amino acid on the next tRNA. So do you want to see what happens? Look, a peptide bond is formed. Okay, and here's what it looks like. Take a look here. This is the big picture. Check it out. Again, we've cut the amino acid off of this tRNA, and now the methionine amino acid is bound to the proline amino acid via a peptide bond. And we have started making a protein, haven't we? This is how a protein is made. Remember, a protein is simply a long chain of amino acids. And finally, we have our first chain. We have methionine linked to proline. We have a peptide bond formed. Isn't that cool? So we're starting our polypeptide chain, and we've got to keep going. We're not done. We have more RNA to read. We have more message to read. And look, this tRNA is now uncharged. You see this tRNA in the P site? Now it's called an empty tRNA or an uncharged tRNA. We don't need this tRNA anymore. We're going to shift everything to the right. The mRNA is going to move 3 codons down the ribosome, right? So everything's going to shift. At this point, this tRNA, this empty tRNA is going to move to the E site. The tRNA here in the A site with the 2 amino acids attached is going to move to the P site. And guess what? The A site is going to be empty. And it's going to be vacant to accept the next charged tRNA. Isn't that neat? And see, again, this is known as the elongation cycle of translation. Again, you have a tRNA in the P site. This tRNA has amino acids attached with peptide bonds between them. If there's an a vacant A site, what happens? The next appropriate tRNA, charged tRNA, binds to the A site. So what do you have? You have the tRNA in the P site. Next to it, you have the fresh tRNA in the A site. At this point, what happens next? Can you beat Wicket? That's right, Wicket. The peptide bond is formed. The P site does its magic Peptidyl-Transferase Activity, which means the peptide chain is broken off of this tRNA and joins onto the incoming amino acid on the incoming tRNA in the A site. This leaves the tRNA in the P site, uncharged. So let's see what happens. You see this? The tRNA in the P site is now uncharged, and the tRNA in the A site now has an extra amino acid. Then remember what I said happens next? What happens next? Can you beat Wicket? That's right. They shift, right? So the tRNA in the P site shifts to the E site. The tRNA in the A site shifts to the P site. And now the A site would have these three fresh nucleotides, this codon in the A site, ready to accept the next tRNA. So look, everything shifts over. And do you know what E stands for? I think you can guess now. Look what's happening to this uncharged tRNA that shifted over from the P site. That's right, the uncharged tRNA gets ejected out the E site. Isn't that neat? And what do you think happens at this point? That's right, the A site is vacant. So the next appropriate tRNA joins the A site, and this cycle repeats over and over again. Every time you finish one of these elongation cycles, the polypeptide increases by one amino acid, and you keep going codon by codon by codon until you have a long polypeptide chain. At some point, you're going to have a long polypeptide chain, and this will continue over and over and over again, extending that polypeptide chain until what happens, do you think? What will bring an end to this elongation cycle? I'll let you think about it for a second, but can you beat Wicket? Right, Wicket. We reach a stop codon. Do you guys remember the stop codons? UAA, UAG, UGA. Any one of those three stop codons will bring a stop to elongation. But remember, elongation can go on and on because these amino acid chains can get quite long, right? Proteins can be hundreds or sometimes thousands of amino acids long, okay? So whenever the stop codon is reached in the A site, right? The A site reads a stop codon. That's when translation is terminated. So let me show you termination of translation. Look at this. At this point, you have a tRNA in the P site, and it's joined to quite a long chain of amino acids. This chain would be hundreds of amino acids long, right? Because you have a properly formed polypeptide protein formed. And at this point, what's in the A site? The A site presents a stop codon. Remember the stop codons? It could be UAG, UAA, UGA. Any one of those three will suffice. Any one brings a halt to translation. And guess what? The stop codons do not have a corresponding tRNA. Remember these structures, these little bluish structures are called tRNA? Well, guess what? There is no tRNA for the stop codons with the anticodon to the stop codons. Instead, a protein. You see this little yellow guy? This is a protein, not a tRNA. It's a protein called release factor. When this protein binds, because it recognizes one of the three stop codons, it actually causes what's called termination of translation and release, right? The ribosomal subunits and other components dissociate. So look, the polypeptide, the protein is free. The large hamburger bun pops off, the small hamburger bun, right? The large subunit of the ribosome, the small subunit of the ribosome, release factor, mRNA, the protein, everything releases because you're done. You don't need to translate anymore. And remember, there's plenty more message on this 3' end of the mRNA. You didn't run out of mRNA. There's still mRNA to read, but you don't read it. You don't translate it because you've encountered a stop codon. Does that make sense? So once a stop codon presents itself in the A site of the ribosome, not a tRNA, but release factor, which is a protein, binds, and it causes release. Everything is hydrolyzed and released. Isn't that neat? And that protein is free to go on and do its job. Remember, this protein doesn't stay a string like this, does it? Do you guys remember? Proteins don't stay. Just because a protein is a long chain of amino acids doesn't mean it stays a long chain of amino acids. Remember, that's just its primary structure. Do you guys remember? Proteins form secondary structures like the alpha helices and beta sheets, right? And then those fold thanks to R group interactions, hydrophobic interactions, disulfide bonds, ionic bonds, hydrophilic polar interactions, right? So these interactions keep the tertiary structure of the protein. So the protein has to fold correctly, right? The protein will fold into its proper tertiary structure. The protein will fold correctly, and then the protein will go and do its job. If this was insulin protein, it'll go off and do its job. It will help your body regulate your blood sugar. Isn't that neat? That's how it works. And remember, once a protein is formed, that protein goes and does its job. Whatever that protein's job is going to give you your traits, right? If that protein's job is to produce pigment for your hair, you're going to have that color hair. If that protein's job is to regulate your blood sugar, it'll go off and regulate your blood sugar. So when we say that genes give you your traits, remember, the genes don't directly give you your traits. Genes code for proteins, right? And those genes need to be transcribed into mRNA, which you can see on this image here. The gene is transcribed with RNA polymerase to mRNA. That mRNA needs to be processed by RNA processing with a 5' cap, a 3' poly A tail, and remember the little introns, which are non-coding regions, need to be spliced out with the spliceosome. At that point, the mature mRNA leaves the nucleus, meeting up with the small hamburger bun, the small subunit of the ribosome. At the 5' end, that small subunit binds to the ribosomal binding site on the mRNA. It then finds the AUG start codon. It also finds the initiator tRNA, charged tRNA, and then the large hamburger bun slams on there. The large subunit of the ribosome attaches. And then there's elongation during translation until you reach a stop codon. And look at here in the background, these aminoacyl-tRNA synthetases are hard at work recharging the uncharged tRNA so that the ribosome always has these charged tRNAs to grab and use. Isn't that neat? So now you know. And when that protein is made, that protein is simply a long polypeptide chain. When that long polypeptide chain is formed, that polypeptide chain forms a protein. It folds into a tertiary structure, and that protein goes on and does its job. That protein goes on and does whatever that protein's job is. If that protein is an enzyme, it will break down its substrate. Again, if that whatever that protein's job is, and that protein's job, whatever that protein does, gives you your traits. Isn't that neat? Isn't that a privilege to understand? You know, I always look at this and I'm fascinated by how amazing and intricate this is and what it really means in the big picture, you know, and how genes give you your traits. At this point, you can actually understand why your genes give you your traits. Isn't that neat? So next, before we finish this chapter, which is the final chapter for my class, Biology 1406, okay, we have one more concept called mutations. So let's jump into that and we'll call it a course. Now, do you remember we discussed mutations a couple times in previous chapters, and I want you to know that a mutation is a change in the genetic information of a cell. So normally what they talk about are mutations inside of genes, mutations in the genetic code. And these mutations change your genes. Think about that. Do you remember that the genes code for proteins? Do you remember that? Genes code for proteins. So if I were to change a gene Do you think that perhaps it would change the outcome of the protein? Because remember the codons, which are 3 letters like CCC or AUG or GGA, right? That's the genetic code, isn't it? And those 3 letters make up a codon. Now, what if I were to change those 3 letters? What if instead of CCC, I changed it to ACC? Do you think that may have an effect on the protein? And that's exactly what happens. If I mutate a gene, if I change the genetic code, if I change the recipe, I'm going to change the outcome, aren't I? So mutations are changes in the gene. Point mutations are changes in just one nucleotide. One nucleotide. If I just change one nucleotide in a codon, that's a point mutation. And there are different outcomes. I want you to know that there are 3 different outcomes that can occur if I change a single nucleotide. And let me discuss those next. Now, these point mutations within a gene can be divided into 2 general categories, and we're going to break these down. The first general category is a single nucleotide pair substitution. This is where you change a G to a C, or you change an A to a C. You change a nucleotide to a different one. The next general category is a nucleotide pair insertion or deletion. This is if you add a letter. You just add an A in the middle of the gene, or you delete a G from the middle of the gene, right? So you add a letter or you delete a letter. So these are the 2 main categories. Let's start with the single nucleotide pair substitutions and discuss the outcomes there. Okay, so nucleotide pair substitutions. Again, this is the category that replaces one nucleotide with another. So there are 3 outcomes that can occur if you substitute one nucleotide for another. One is a silent mutation outcome. The next is a missense mutation outcome, and the last is a nonsense mutation outcome. So let's talk about a silent mutation. A silent mutation is when you substitute one nucleotide for another, but this has no effect on the amino acid produced by a codon because of redundancy. So let me give you an example. For example, take a look. What if I have a codon that should read CCC? The codon should read CCC, which codes for the amino acid proline. Now, what if I mutate that to CCA? What would happen? Take a look. Well, when I look at the codon table, CCA is different because I've mutated CCC to CCA. Does that affect the outcome of my protein? Does my protein get built differently? No, because CCA codes for proline, CCC coded for proline. So even though I mutated the DNA from CCC and I substituted this final C for an A, it did not have a deleterious effect on the protein because the same amino acid was added. So there was no real effect on my protein. This is known as a silent mutation. Silent mutations occur when the mutation changes the gene, but it does not change the amino acid it codes for, and therefore it should have no effect on the protein. It should have no effect on the protein that's created. So again, what is a silent mutation? It is when the gene mutation results in the same amino acid produced, and this is due to redundancy. Remember, this occurs because there's more than one codon that codes for the same exact amino acid. All right, so now let's talk about a more damaging type of mutation. This is known as a missense mutation. A missense mutation is when you change a letter, the codon to still code for an amino acid, but not the right amino acid. Let me give you an example of a missense mutation. So take a look. What if I have CCC, right? And I give it a substitution from CCC there's a point mutation substitution to ACC. So I just changed this first C to an A, right? So it's ACC. Well, CCC should have coded for proline, the amino acid, but guess what? ACC codes for threonine. Isn't that interesting? So that's known as a missense mutation. I changed one nucleotide, and I got a different amino acid. Now, lastly, what is a nonsense mutation? This is a scary one, okay? This is when you change an amino acid, okay? You change an amino acid codon into a stop codon. This is really bad because usually this leads to a non-functional protein. So again, a nonsense mutation changes an amino acid codon into a stop codon. So let me give you an example here. All right, check this out. What if I have a codon, CAA, right? This is the normal codon in the gene, CAA, which codes for glutamic acid, or Q. And instead, it gets mutated from CAA to UAA, or on the DNA, it would be TAA. So it would be UAA. What's UAA? UAA codes for stop. Isn't that terrible? So in this case, the ribosome, instead of reading CAA and putting glutamic acid and moving on to do the rest of the protein, it thinks it's done making a protein. It's going to make what's called a truncated protein or a shortened protein. And usually this is a non-functional protein. So this is really bad, right? Because you're introducing a premature stop codon into the protein. And so you're going to end up with a protein that's way smaller than it needs to be. And if that's the case, it's probably not going to function correctly. Isn't that weird? So again, silent mutations, you mutate the gene, but you don't change the protein. You don't change the amino acid outcome. Missense mutation, you change the codon and you change the amino acid it codes for. Nonsense mutation, you change the codon and you change it from an amino acid to a stop codon, leading to a premature stop codon, right? Well, and sometimes this could be well in advance of where the stop codon should be. That could be potentially really deleterious. So anyway, these are all known as nucleotide-pair substitutions. Do you remember when we discussed sickle cell anemia and I told you that in sickle cell anemia, there was a mutation that resulted in a glutamine or glutamic acid residue, a glutamic acid amino acid being changed to a valine amino acid. And remember, that was the whole reason that the sickle cell phenomenon happened. The sickle cell phenotype occurred. Let me ask you this. Can you beat Wicket? What type of substitution mutation is this? Is it silent? Is it missense? Or is it nonsense? That's right, Wicket. It is a missense mutation, you guys. Look at this. Now you understand it in so much detail. Remember we talked about sickle cell. In sickle cell, there is a codon, GAG, which codes for glutamic acid. Now, that codon has been mutated to GTG. See, it's a substitution mutation. And GTG codes for the amino acid valine. Isn't that interesting? So yes, now you fully understand sickle cell, right? Sickle cell is an example of a missense mutation. The simple change of an A to a T changed the amino acid remember, it was the 6th residue or the 6th amino acid of the beta hemoglobin gene. And that's what resulted in the misfolding of beta hemoglobin, exposing that hydrophobic region, causing the different hemoglobin proteins to stick to one another and concatenarize, which means stick to one another, forming those filaments that stretched out the cell into a crescent shape or a sickle shape. Isn't that neat? So isn't this a privilege, everyone, that you understand how mutations work? That mutations result in changes in the primary structure of a protein, which can affect the tertiary structure of the protein. And as I've been saying all along, you guys, if a protein does not fold correctly, it does not function correctly, and this can result in problems, right? This can result in traits like sickle cell. Next, let's talk about insertions and deletions. What are insertions and deletions? And remember, we're just talking about point mutations. So for insertions, we're adding a nucleotide, and for deletions, we are removing a nucleotide from the gene. Now, this is terrible. This is really terrible because these mutations have a disastrous effect on the resulting protein more often than substitutions do. You know why? Because they cause what's known as an altered reading frame, producing a frame shift mutation. Think about it. You're shifting your reading frame. So in this case, by adding a letter, remember, the ribosome is reading every 3 letters, right? The ribosome is reading every 3 letters, every codon. If you were to add a letter, you're going to shift the reading frame over. And so everything from the frame shift mutation onwards, everything from the insertion or the deletion onwards is going to be out of frame. So every single codon is going to be wrong. Okay, and I'm going to give you an example of what happens when you have a frame shift mutation. Now, let me just give you a sense of the chaos that can ensue if you introduce an insertion mutation into a codon. Here I have something that, resembles codons, but it's obviously English. The cat ate the hat, right? So this is obviously in English, but you can imagine I'm trying to make a point where every 3 letters represents a codon, right? And remember, the codons code for different amino acids. Right now, imagine if I were to do a point insertion. Watch this. What if I added an extra A right here? Like I added an A between this C and this A in cat. So look at this. I added this A. Now look what happens. The ribosome reads the correctly, but when it reaches cat, because of the insertion, the rest of the sequence is shifted. This is what's known as a frame shift or a reading frame shift. Your reading frame went from 3, 3, 3, 3, 3, right? The cat ate the hat. Your reading frame is now shifted, right? The ka tat ith iha, right? You see how everything after the insertion becomes gibberish? That's because you have moved your reading frame over, okay? And when you move the reading frame, everything becomes gibberish. Everything becomes gibberish. Before it made perfect sense. The cat ate the hat. But now everything is gibberish. So every codon from the insertion point onward becomes incorrect. And this is highly deleterious. This is terrible. This will result in catastrophic protein, problems where the protein is not created correctly. The protein obviously will not function correctly, and that protein's function will be lost. Same thing with deletions. Look at this. Imagine if I deleted this C. Imagine if there was a point deletion where this C got deleted. Now look at this. Again, same thing. There's a frame shift. The A shifts over, and again, the phrase makes no sense whatsoever. The ada, tet, he, at doesn't make any sense. Total gibberish. So again, insertions and deletions are catastrophic for gene expression. And they are usually much, much worse than substitution mutations of the same kind of point variety. So that leads to the end of the chapter, you guys. Can you believe we reached the end of the final chapter for Biology 1406, Biology for Science Majors 1? I hope you learned a lot. I hope it was a good experience. Please let me know. Share your experiences in the comment box below. I love reading them. I haven't been the best about responding because I'm so busy making these videos, but I'm going to be spending, now that I have free time, I'm going to be answering your comments, and I'm going to be getting back to you more. Thank you so much for tuning in. It means a lot to me, especially if you're not one of my own students. I've had students around the world reach out and tell me that, they appreciate the way I explain things, and I, help them to pass their classes. And that just makes me so happy and brings so much joy to me and my life. So thank you so much. I hope you took away a lot. I hope you learned a lot, and I hope you're prepared for your next semester. And I wish you all the very best in the future. Stay in touch, and until next time, I'll see you guys.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.